All right, good morning, everyone. Let me start off the day with something that's a little bit stressful and depressing. Remember, a week from today is the second exam for this class, okay? For those of you who are taking the online version of the class, we'll have you go to the same room you went to the last time. That's 101 Fawcett. For those of you who are taking the not online version of the class, you should be coming here to 120 Med Sciences, right? Where you're sitting right now, I presume. 45 questions, all multiple choice. Looks a lot like the exams that you'll find on the pilot page for the course. If you haven't looked at those exams from previous offerings of the course, please look at the exams from the previous offerings of the course. That will be a great way to help you focus on what sort of questions I'm going to ask and the way that I'm going to phrase those questions. If I haven't talked about something in lecture, I'm not going to ask you about it on the test. All right? So if you find you're looking at some of the old exams, particularly some of the older exams, like when before Wright State was on the semester system, if you find you're looking at those old exams, you say, gee, I don't know what this is about, where'd this come from, did this get talked about? If I didn't talk about it for this class, I'm not going to ask you about it on the test, okay? When you're done with the test, go home, right? There's no lecture or anything after the exam is done. You'll need a number 2 pencil to be able to fill in those bubbles again. And there will be an answer key posted on Pilot shortly after the exam's over. And we'll have a hard copy of the answer key around here at the front of the room for you to look at too on your way out if you really like to. All right, enough said about that. I'll bring it up one more time on Tuesday, but again, a week from today, second test for this class. Okay, so let's just get right back to work then, now that that unpleasantness is out of the way. In lecture on Tuesday, I talked with you about how it is that eukaryotic cells reproduce, and really very specifically how eukaryotic cells solve one particular problem, and that's the problem of replicating their nucleus, including replicating the stuff inside of the nucleus, the DNA. Living cells invariably come from other living cells. I didn't shout that out to you on Tuesday, but you can't make a cell unless you've got an existing living cell to do that business of cell division, and again, for eukaryotes to do the business of mitosis, replicating the nucleus. By the same token, you can't make an organism unless you start with an existing organism. So in Tuesday's lecture, we talked about how cells divide. The thrust for today's lecture is certainly related, but it's also about how it is you make whole new organisms, and particularly from the perspective of eukaryotes, where most of cell division, where most eukaryotic organisms come as the result of sexual reproduction. So today, I want to take some time and talk with you about how it is that we solve this additional problem, not just a matter of cell division, but also a matter of creating new organisms through a process that's ultimately additional cell division, and then talk a little bit at the very end about why it is this is such an advantageous system. To put it very bluntly, why is it advantageous to have sex? I'm sure that many of you, if not all of you, already have some ideas about why it is that sex is a good thing. I will tell you from a very clinical biological perspective, from a genetics perspective, about why sex is such a huge advantage to organisms that have the ability to participate in sexual reproduction. All right, so that's where we're going, my big finish. I'll tell you why it is that sex is good. All right, but before we get there, let's do a little bit of work and talk about what's going on here. So today, I'm going to talk with you not so much about mitosis. I'm going to talk with you about this other thing called meiosis. So the first word on the lecture outline today is really what I'm going to talk with you about today. Let me remind you, mitosis, in a word, that's the word we use to describe nuclear replication for eukaryotic cells. We can break it down into smaller parts. I did that how many times? 5, 6 times, over and over again in lecture on Tuesday. I told you it starts with prophase. You get to an instant in time called metaphase. You move from there to another place called anaphase. You wrap it all up with telophase, and you can start the full process over again if you like. What had to happen during mitosis? Well, you need to break apart that nuclear envelope. That starts to happen in prophase. You need to condense the chromosomes. Starts to happen in prophase. You need to get the mitotic spindle set up, and so the centromeres start to move to opposite sides of the cell. That's how you start. That's what you need to do. That's what's going on in prophase. When the chromosomes are as condensed as they can be, when the mitotic spindle is all set up, the centromeres, the centrosomes are on opposite sides of the cell, the nuclear envelope is completely gone, and all the chromosomes are lined up in a row. That's metaphase. That's like the midpoint in the process. And then after that, it's just a matter of getting the chromosomes to opposite sides of the cell, so the mitotic spindle shortens. The chromosomes don't need to be condensed anymore, so they start to decondense. The nuclear envelope starts to form around the clusters of the chromosomes as they're moving apart from each other. That begins in anaphase and it ends in telophase. By the time you're done with telophase, the nuclear envelope is completely back. The nucleolus shows up again. It had disappeared during prophase. The chromosomes are decondensed. The mitotic spindle is gone. You're back to where you started. Right? So what had to happen? You had to get rid of the nuclear envelope. You had to separate the chromosomes, and you had to get them to opposite sides of a cell. That's what you got to do. That's the mission. How do you do it? I'll put it to you. I'll suggest to you, you do it in the most straightforward, most logical way you possibly could. All right? So if you're trying to wrap your head around what's going on during mitosis, I suggest you do it by thinking about what needs to happen. Maybe a little bit about why it needs to happen, but if you focus on what needs to happen to get the job done, it just seems to follow logically from that point. And then the rest is just a little bit of memorization of vocabulary words like, oh, prophase, metaphase, anaphase, and telophase. The Derby's starting early today. They're a little ahead of schedule. Okay, so that's what mitosis was about. That was all about making 2 nuclei when you start out with 1 nucleus. Okay? Today, we're talking about something similar but different. Sounds similar, but is fairly interestingly and significantly different. Today, we're talking about meiosis instead of mitosis. And what happens in meiosis is where now the focus is taking a nucleus, and whereas you start with a nucleus, you end with 4 gametes. Okay? For mitosis, you started with a nucleus and you end with 2 nuclei. For meiosis, you start with a nucleus and you end with 4 gametes. And gametes is fancy talk for sperm and eggs in our parlance, right? For plants, it would be pollen and ovum, right? But come on, we're not worried about pollen and ovum nearly so much as we're worried about sperm and eggs. Sperm and eggs are the kind of gametes that animals are making. How you make them is ultimately through this process called meiosis. So you start with a nucleus, and from that 1 nucleus, you end up making 4 gametes. So that's what's going on, and that's what needs to be accomplished here. And a big part of the problem, of course, what are you going to do with those gametes? You're going to have sexual reproduction. Do you need me to tell you? An egg hooks up with a sperm, and then you create a new organism. It can undergo lots and lots of mitoses and make a whole big new organism after that. Okay? So we're making eggs and sperms. We're making gametes. That's what has to happen. the thing that's kind of like the reason that you have to do it with meiosis as opposed to mitosis is by going from one cell to 4 gametes, what's going to happen is you're going to correct a problem that you would have otherwise had associated with ploidy. I've been stammering for the last 30 seconds trying to think, how am I going to spit out the idea of ploidy? Ploidy is what we use to describe the number of copies of genes within an organism, the number of copies of chromosomes within an organism. I got news for you. Probably you've heard this before. You, as a human being, are a diploid organism. Diploid means that inside of all of your cells that have nuclei, inside of all of your cells, there are 2 copies of all of your genetic instructions. You knew that, right? One of those copies comes from your mother. One of those copies comes from your father. Your father's copy is one ploid. Your mother's copy is another ploid. You got 2 of them, di, but it's diploid. You got 2 copies of your genetic instructions. Human beings are diploid organisms. Gametes, eggs and sperm, are not diploid. They are haploid. They have only 1 copy of all of those instructions. And what happens is by using haploid gametes that fuse together to become a diploid cell, specifically a zygote, a fertilized egg, by having haploid gametes come together to make a diploid cell, you're preserving the ploidy for the organism. The alternative would be something like this, right? One of your cells, diploid, bumps into somebody else's cell, diploid, all right? And what do you get when you fuse together 2 diploid cells? You don't have just 2 copies of all of the chromosomes. You're going to have 2 from the first person and 2 from the second person. Look to your neighbor, right? They're giving you some of these chromosomes, right? You're not going to have just 2 copies. Yeah, you 2 guys I'm worried about. Yeah, you're going to end up with 4 copies, right? We got a word for cells that have 4 copies of the chromosomes. That's called tetraploid, tetra, 4, right? That's not so good. I suppose it could work out for a while, right? Having 4 copies of the chromosomes inside of the cell instead of 2. But what's that progeny? What kind of problems is that progeny going to have? They've got to find a nice, receptive, also tetraploid individual to bump their cells into and end up making an octoploid, right? It's getting out of hand pretty fast here, right? The octoploid folks are going to have to find other octoploids to make whatever the heck you call a 16-ploid, right? And it just gets out of hand real fast. And so what's happening with meiosis, by making haploid gametes, is we're reducing the copy of the number of we're reducing the number of copies of chromosomes that we have so that we can maintain existence and maintain organisms that are diploid all the way through. That's the crux of the problem here. We need to adjust and reduce chromosome number if we want to go through the process of sexual reproduction. So ploidy. Now, here is a schematic that gives you a sense of what's going on with human reproduction. Nothing going on here aside from the fact that I get to flash some gratuitous nudity at you, and you've got a diagram that's showing you what I've just been saying about reducing ploidy number. We live most of our lives as human beings as these fancy diploid organisms down here, but once in a while, some of our cells, in women, ovaries, in men, testicles, testes, undergo this special process of meiosis to make haploid gametes, sperm and eggs. These haploid gametes, shown here with the blue path as opposed to the salmon-colored path, they come together, they fertilize, an egg becomes fertilized, it's a diploid zygote, and through mitosis and development, you end up with these fancy organisms that can do the whole process over again. Okay? Let me point out to you, that within our bodies, yours and mine, there are two different kinds of cells that biologists recognize as being a really important difference. There are the cells that can undergo mitosis, and more importantly, perhaps, the cells that can do both mitosis and meiosis. Our muscle cells, our skin cells, our brain cells, our blood cells, our liver cells, our heart cells, pretty much all the cells that we think of as being really important and central to who we are the cells that only can do mitosis. Those are called somatic cells. Cells that can only do mitosis are somatic cells, and that's in contrast to the other kind of cells that can do both mitosis and meiosis. We call those germline cells. Germline cells, in this diagram here, would be called either ovaries or testes. Appreciate the difference. These cells can make more copies of themselves through nuclear division, this whole business of mitosis I talked with you about on Tuesday. But they also can, in addition to making more copies of themselves, can sometimes go through this meiosis process that lets them make some gametes. And that has, again, from a biological perspective, for biologists, this turns out to be a really fundamentally important difference. If you get a mutation to one of your skin cells, that could be bad for you, right? That mutation could ultimately give rise to a tumor, uncontrolled cell growth, it takes over your body, and you die, right? Not a happy thing for you, okay? But the only mutations that can get passed on to the next generation, the only mutations that can get passed on to a subsequent generation are those that happen in germline cells. The consequences for a mutation in a germline cell are actually very different than the consequences of a mutation in a somatic cell. Somatic cell mutations are bad news for you. Germline mutations actually probably don't affect you that much, but they're going to have an impact on the rest of your lineage from that point on. Those are the sorts of things that can get passed on. So from the perspective of somebody studying evolution or population genetics and how it is that gene frequencies change and how species adapt to their changing environment, it's the mutations that are taking place in the germline cells that are where all the action is, and that's where the focus is, as opposed to the things that you and I as individuals worry about. And that actually gives rise to a sort of interesting mindset about people who think about these things a lot. They start talking as if all the stuff that you and I are so focused on and proud of about our bodies, oh, what a delightful hair color I have, right? Oh, look how tall I am. Look how smart I am. All the sorts of things that we think are pretty swell about ourselves turn out to just be a vehicle, from this perspective, to just be a vehicle to keep those germline cells safe and warm and in a position where they might be able to bump into other people's gametes to give rise to more germline cells that can go on. So there's a school of thought, a lot of folks within biology reduce us all down to just our germline cells. Everything else is window dressing, at least from the perspective of the species and propagation and making more individuals and adapting to our environment. Okay, so can you see how somebody might get that mindset? Again, it's kind of an interesting way to look at things. Our whole bodies are simply there to keep our testicles and our ovaries in a position where they may be able to carry out this process. And we may think all the rest of what we do, we're proud of all the rest that we do. You could argue that it is simply there so as to further advance the causes of our ovaries and our testicles. All right, so there you go. That's the cycle that we have to go through. We live most of our lives as diploids. We make these haploid cells that can bump into other haploid cells and become diploid organisms, and you can start the whole process over again. The next couple slides are simply going to point out to you that that's the way that animals, and particularly humans in this picture, but animals have chosen to play this game. I want to open up your mind here. I just want to make sure you appreciate that there are other ways that this game could be played. The core principle of the game is that there's a diploid stage of life and there's a haploid stage of life. For organisms that are sexually reproducing, that's the two phases of the life. There's a part of the life that you live as a diploid cell. Part of the life history of the organism is as a haploid cell. For animals, like you and I, most of the emphasis, most of the time is spent as a diploid organism. But it doesn't have to be that way to still conform with this rule for sexual reproduction. So animals, like us, take this path. There are alternatives. So let's talk about these a little bit. So move away from the nude man and woman. There's a mouse. Mouse is an animal just like us. Most of a mouse's life is spent as a diploid organism, but they have germline cells that can undergo meiosis. When those germline cells undergo meiosis, they make gametes, haploid, eggs and sperm. Those can come together to make diploid zygotes, and you can do the whole process over again. Most of the life, though, is spent, the most abundant, the most obvious organisms that are mice are not, when's the last time you guys noticed a mouse sperm? Right? Doesn't come up that much. When's the last time you noticed a mouse? This time of the year is when they're coming in from outside to find a warm place to live for the winter. If you're living in Wright State housing, keep your eyes peeled, and you may well find some opportunities to see mice later this afternoon, if you're looking carefully enough. Right? You don't see the sperm of these organisms or the eggs of these organisms. You see the organism because they're spending most of their life, the most substantial impact they're having on their environment is from when they're a diploid organism. That's the strategy that you and I have as we're playing this game. That's what other animals are doing when they're doing sexual reproduction. The gametes are somewhat insignificant in the greater scheme of the impact they're having relative to the diploid organism. But here's the thing. There are other organisms that have adopted a different strategy, where the haploid organism takes on a greater role, a greater significance. So there are some plants and algae that spend relatively equal amounts of time as both the diploid organism and the haploid organism. And they're often very similar in appearance. And then check this out. There's most fungi and a few free-floating around protist kind of organisms actually do the reciprocal of what you and I do. The most prominent organisms that you find in the environment, the ones that are having the biggest impact on the world, are not the diploids like you and I, but rather the haploids. And the thing that's fairly insignificant, transient, you don't see it that often, only under very special circumstances, are the zygotes that are diploid. Okay? You have to pass through this phase to be able to give rise to the haploid cells that, again, become the predominant organism within the ecosystem. But it's the diploid guys that are somewhat marginalized and the haploid guys that are most prominent for the organism. So that's what I wanted to tell you about life cycles. The key is this, right? You pass for sexually reproducing organisms. There are times that you're diploid, and then there are times that they have to be haploid, and you just go in a circle around that. Different organisms have adopted different strategies regarding, do they focus their energy, is the organism most prominent when they're diploid, or is it most prominent when they're haploid? Obviously, for animals, we're focused primarily on the diploid part. All right, so let's talk about meiosis. Once again, mitosis. We started with a nucleus, and we ended up with 2 nuclei. Okay? We're moving on. Meiosis. We start with a nucleus, and we end up with 4 gametes. Okay? So let's talk about how we pull that trick off. How do you start with a diploid cell, and you end up with 4 haploid gametes? To get there, let me take at least a minute or two to get us all on the same page with some vocabulary. So this turns out to be a fairly helpful and important slide, and it's going to lay the foundation, I hope, for a lot of the pictures you're going to see in the next couple of slides, the next several slides for today's lecture. So let's start up here at the top left, and let me draw your attention to some things that I think will help you keep track of what's going on here. For the rest of today's lecture, you're going to be seeing some chromosomes that are shown in red and some that are shown in blue. All that they're trying to do is draw your attention to the fact that as a diploid organism, you get half of your chromosomes from one parent and half of your chromosomes from another parent. Half of your chromosomes come from, for just the purpose of illustration, a red parent, and half of them come from a blue parent. And they take it a step further here, and they tell you that the red chromosomes are the maternal, they're coming from the female parent, and the blue chromosomes are the paternal, they're coming from the male parent, right? Please do not begin to think for a moment that in a microscope a male's chromosomes look blue and a female's chromosomes look red. This is just to help you. It's a scorecard. It's a device to help you just visually keep track of where the one set of chromosomes are relative to the other set of chromosomes. All right? But that's going to be a convention you're going to see being followed for a whole bunch of slides yet here today. Red ones come from one parent, happens to be the mother. Blue chromosomes come from another parent, happen to be the father. They're showing you a cell here. And if you want, you could say this is a nucleus, right? If it is, then the rest of the slide's a cell. But they're showing you an organism, and they're giving you a complete look at all of their chromosomes. There's a fancy word for that, actually. When you're looking at a picture that shows you all of an organism's chromosomes, that's called a karyotype. The very last slide I have for you today shows you a human karyotype. You just get to see laid out the complete set of a human individual's chromosomes. Okay? What you're seeing here, we could call this a karyotype, but what you're seeing here is this, is that, well, let's talk about it. You've got 3 different red kinds of chromosomes and 3 different blue kinds of chromosomes. Okay? It happens that the blue chromosomes are present in 2 copies each, right? There's a big blue one here. There's one big blue one and there's one big blue one. There's 2 big blue ones. There's 2 medium-sized blue ones. There's 2 small-sized blue ones. So not only do we have a big blue and a big red, we actually have 2 big blues and 2 big reds. This is actually a tetraploid cell. So if I count the number of blue chromosomes, I'm sorry, let's do it this way. If I count the number of big chromosomes, I see 1, 2, 3, 4. So this is, strictly speaking, what we're looking at right now is a tetraploid cell. This organism happens to have 3 different chromosomes. And so their chromosome number, N, is 3. There are 3, well, there's a big one, a medium-sized one, and a small one. Okay? So there's 3 different kinds of chromosomes. If this was humans, you wouldn't be seeing just 3 chromosomes. You'd see 23 chromosomes. Our chromosome number is 23. We have 22 autosomes and 1 sex chromosome, the X and the Y chromosome. Okay? So we have 23 pairs of chromosomes. Whatever the heck this thing is, it only has 3 pairs of chromosomes. Okay? So it's got 3 chromosomes. Oh, and I'll tell you this too. I've mentioned this already in passing. If this was a human, the convention would be this. The big chromosome, the biggest chromosome, you'd call chromosome 1. The next biggest, you'd call chromosome 2. The next biggest, chromosome 3, and so on. So if you like, if it helps you keep track of what's going on here, instead, because I'm stumbling over saying big and medium and small, maybe I should just call the big one chromosome 1, and the medium one chromosome 2, and the small one chromosome 3. I may opt to do that at some point here. All right, so this organism, it's got genetic information it got from a male parent, genetic information it got from a female parent. It's got 3 different chromosomes, so its N number is 3. 2 times N is 6, right? So the diploid cells would have 6 chromosomes, but what you're actually seeing in this picture is not a total of 6 chromosomes, but actually a total of 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So it's not 2N, it's actually 4N. This is a tetraploid cell, as it's being shown to you. If there was only one big blue and one big red and one medium and one, it would be a diploid cell. What you're seeing here is that the big blue one is actually present in 2 copies. And how did you get the second copy? Well, you got the second copy because of DNA replication. That chromosome got duplicated. We'll talk about how at a molecular level that gets done in another week or two. But you started out with one blue chromosome, big blue one, and you made a copy of it. So we've got 2 copies of this particular chromosome. For meiosis, they're going to stay stuck together at this constriction point here called a centromere. So you've got the 2 copies of the chromosome remain tethered together at a centromere, and we call each of those copies a chromatid. And specifically, we call them sister chromatids because this one is a perfect copy of this one. So not only is this a chromatid and this a chromatid, but they're actually sister chromatids because they have exactly the same nucleotide sequence, exactly the same set of genes. That's a little bit different than what you're seeing going on over here. Here, we've got a blue medium-sized chromosome. Here we have a red medium-sized chromosome. This is a chromatid. That's a chromatid. These are sister chromatids. These are similar, but not necessarily exactly the same. So those get called non-sister chromatids. It's a tip of the hat to the fact that in general, they've got the same set of genes and the same set of genetic instructions, but they're not necessarily identical to each other. Sister chromatids are identical to each other. Non-sister chromatids have the same lay of the land, the same general organization of genetic information, but there may be differences between them in some of the particulars here because they came from different parents. And what we're talking about here, because these are chromatids that have the same sequence of genes, the same general genetic information, we call those homologous. Right? Homologous is fancy English for the same. They're homologs. One's roughly interchangeable with the other. They're pretty close to being completely interchangeable with each other. So any copy of chromosome 2 for this organism would be a homologous chromosome to any other copy of chromosome 2 for this organism. All right, so again, this slide has gotten boring. I appreciate that, but there's some vocabulary, and I just want to make sure we're all seeing the same things, myself included, as we go through the rest of this. Because here's what's got to happen. All right, the big picture is we're going to start out with a diploid cell. They've simplified that previous cell for you, so now all you're seeing is the big chromosome. No more 3, there's just 1 chromosome that we're focused on here. You start out with a diploid cell. 1 copy from the organism's father, 1 copy from the organism's mother. These are homologous chromosomes. Okay. The DNA gets replicated, and you end up with these sister chromatids that remain stuck together. Then, this is all going on during interphase. Then, in meiosis, what's going to happen is you're going to have 1 round of nuclear division where you put into 1 cell 1 of the pairs of sister chromatids, and you put into the other cell the other pair of sister chromatids. Those sister chromatids still stay stuck together. This is meiosis. This is how we're going to make gametes out of this 1 diploid nucleus that we started with. Excuse me. It was diploid. A round of DNA replication or synthesis takes place to make you something that's tetraploid. This tetraploid cell undergoes 1 round of nuclear division to give you 2 diploid cells. So this is looking sort of like mitosis right now, right? The only real difference is the sister chromatids are still stuck together. And then you go through 1 more round of nuclear division without an intervening round of synthesis. So you take your two diploid cells and you have them undergo one more round of nuclear division. There's no more, the DNA doesn't get replicated here the way it would have in mitosis, and you end up with 4 haploid gametes. That's what meiosis is all about. You start with a diploid cell, you have one round of DNA replication, and you have 2 rounds of nuclear division, and you end up with 4 gametes. Start with a diploid cell, you end up with 4 haploid gametes. Okay? So that's meiosis in a nutshell. And let me talk with you now in a little more detail about exactly how it is this is playing out. So here's the whole shebang, and guess what's going to happen? In the next couple of slides, we're going to focus our attention on different segments as we move across this whole spectrum of what needs to go on. You start with a diploid cell, you end up with 4, and this isn't even the whole story apparently, you end up with 4 haploid gametes. See the coloring going on there? You got the blue chromosomes, you got the red chromosomes. With mitosis, there's prophase, metaphase, anaphase, and telophase. You know somebody's trying to talk with you about meiosis when instead of saying prophase, they say prophase 1 or prophase 2. Because what's happening with meiosis is you're doing the same things that you're doing in mitosis, it's just you do them twice. The first time you do them, it's called prophase 1, for instance, and the second time you do prophase, that's prophase 2. So check it out. We're starting here with prophase 1, and what do you suppose has to happen in prophase 1? What has to happen in prophase 1 is pretty much in meiosis is pretty much the same thing that has to happen with prophase in mitosis. You got a pesky nuclear envelope. You got to get rid of it, right? You got long strung out chromosomes all tangled together. You'd better start condensing those chromosomes. And you also better start laying the groundwork that's going to allow you to pull sister chromatids to opposite ends of a cell. That's a mitotic spindle in mitosis. Guess what? In meiosis, it's called a meiotic spindle because this is happening in meiosis instead of mitosis. What's happening at a molecular level is actually the same. And the reason for it is the same. It's just now this is going to be taking place in a germline cell. And this germline cell has begun to go down the path to making haploid gametes. Same thing. So if somebody were to ask you, what's going on in prophase 1 of meiosis? It's the same answer I think you would give if you were talking about prophase of mitosis. There is something a little different. And we'll talk more about this after we get through what's actually going on in meiosis. In prophase 1 of meiosis, there's this other thing that's going on called recombination or crossing over. This doesn't happen in prophase of mitosis. It only happens in prophase 1 of meiosis. And I mean that fairly emphatically. It only happens in prophase 1 of meiosis. And again, we'll talk more about that in about 10-15 minutes. You can see this taking place with a light microscope, the crossing over. Literally, the chromosomes lay on top of each other, and they look like an X. And somebody who must have had a good religious training, some sort of Christian religious training early on, about 150, 200 years ago, called that crossing over a chiasmata. Okay? Chi is the Greek letter. It looks sort of like a cross, right? They were talking specifically about the fact that it looks like a cross. It's got the Greek letter chi there. There's crossing over that's taking place. Okay? Well, again, we'll talk more about this later, but that's why this new word is there. So after prophase 1, what's got to happen? Right? You got to condense your chromosomes, got to get rid of the nuclear envelope, you got to get the mitotic spindle or here the meiotic spindle set up. After that's accomplished, The chromosomes get to be as condensed as they can be. They line up on a plate in the middle of the cell, and the spindle starts to pull them apart. It's set up so that it can pull these chromosomes apart. What did you call that in mitosis? That's called metaphase. What do you call it in meiosis? That's called metaphase 1, for at least the first time that it happens. After metaphase, in mitosis, you went into anaphase. Guess what? In meiosis, you go into anaphase. And what's happening in anaphase? Here, the meiotic spindle begins to shorten, pulling chromosomes to opposite sides of the cell. Eventually, the nuclear envelope starts to reform around these clusters of chromosomes. The chromosomes become less condensed after you've finished metaphase. And there's one kind of interesting difference that's taking place here with meiosis that we didn't see taking place in mitosis. We saw this business of crossing over. That doesn't happen in mitosis. Another thing that doesn't happen is that in anaphase and then beyond for meiosis, the sister chromatids remain connected to each other at their centromere. So in mitosis, those centromeres would have fallen apart at metaphase, and you would have pulled apart the chromosomes to opposite sides of the cell. Here, the copies of the chromosomes, the one chromosome that got made as a copy of the other chromosome, those sister chromatids remain tethered together, physically attached at their centromere after this round of nuclear division. So they get pulled apart as a pair. And one cell will get the blue copies. One of these gametes will get the blue copies of this chromosome. Here, look at the little guy there, right? One of the gametes will get the blue copies of the little blue guy. The other cell will get the red copies of the little red guy. Okay? It was because they stay tethered together. They're attached at their centromeres still. When you're done with this round of division, what do you got? You've got chromosomes that aren't very condensed. They're wrapped up in a nuclear envelope. You've got no more spindle involved anymore. You get cytokinesis, a cleavage furrow that separates the two things, the cell into two smaller cells. And what do you got? You end up with where you started with a tetraploid cell. You end up here with a pair of diploid cells. These diploid cells are a little bit unusual because the sister chromatids are still connected together, but otherwise it looks for all the world as if mitosis has just happened. Hey, I see a pattern developing here. It happened to us on Tuesday. It looks like I set myself up for the same sort of thing to happen here when I put these slides together. Check it out. This is a zoomed-in version still of the zoomed-in version. Look what's just happened to you here. We started with this zoomed-out version, and then we zoomed in on this. And now I'm going to have you zoom in on this, all right? The first two in the process. I think I've said enough about this at this point. So why don't we move on to the next two? And again, remember the end product here of this first round of meiosis is a diploid cell, a pair of diploid cells. Only thing that's different is the sister chromatids remain connected to each other at their centromeres. All right, you tell me what you would like, because I think you can predict what's happening here. All right, would you like for me to just prattle on about what goes on for the whole thing, or do you want me to focus on these two and then on these two? Why don't we zoom in and do the focus this time? But bear in mind, this picture's showing you what's going to happen. Let's take a couple minutes to talk about what happens here, and then a couple minutes to talk about what happens here. How about that? So, let me remind you, you started with a diploid cell. That diploid cell underwent a round of DNA replication to become a tetraploid cell. That tetraploid cell went through one round of meiosis, meiosis 1, the first part of meiosis. It did prophase 1, metaphase 1, anaphase 1, and telophase 1. At the end of telophase 1, we now have two diploid cells. Okay? Started with a diploid cell. It became tetraploid for a little while. Now we've got 2 diploid cells. We're only halfway there because we need now to reduce the chromosome number so that we end up with 4 haploid gametes. As prophase 2 begins, notice, remember, there is no more DNA replication that's taking place. We have diploid cells, and we're going to end up making haploid cells. Unlike what would have happened in mitosis, you don't replicate the DNA here. Okay? So there is no interphase that involves a DNA replication. Instead, you go straight from having these 2 diploid cells at the end of the first round of meiosis to the second round of meiosis, and we start off with prophase 2. And guess what? You do the whole dance all over again. The only thing that's different is you didn't do a round of DNA replication. And these sister chromatids are still stuck together from the first round of meiosis. Okay? So you do it all over again. Goodness knows I'm sick of saying it. You've got to be really tired of my telling you. What do you got to do? You got to condense your chromosomes because they're all loose and tangled up with each other. They're all bundled together with a nuclear envelope. Get rid of that thing. You're going to need to set up some sort of spindle, some set of tubules that are going to let you pull apart chromosomes to opposite sides of the cell. Why don't you get to work on getting that set up? That's what prophase 2 is all about. It's the same story as what was going on with prophase and mitosis. It's the same story as what's going on with prophase 1 in meiosis. We're going to do that whole dance one more time. Nuclear envelope starts to go away. The chromosomes begin to condense. They had decondensed at the end of telophase 1. They're condensing again, now in prophase 2. And the centromeres are moving to opposite sides of the cell. They have replicated. The DNA has not. They're moving to opposite sides of the cell so you can start pulling these chromosomes to opposite sides of the cell. At the end of prophase 2, guess what? The chromosomes are as condensed as they're going to get. The centromeres are on opposite sides of the cell. The meiotic spindle is all set up again. The chromosomes are all lined up on a plate. You guys know the word for that. That's what we call metaphase. It happens here because it's the second round of meiosis. We're going to call it metaphase 2. And it's happening simultaneously in both of these daughter cells. What do you suppose happens after metaphase is over, after metaphase 2 is over? Does the picture need to even show up for you to be able to predict what's going to happen? I don't think so, because guess what's going to happen? We're going to separate those 2 sister chromatids. They lined up together on the metaphase plate, but now, unlike the first round of meiosis, we'll separate the sister chromatids. They'll no longer be held together at their centromeres, and those separated sister chromatids will get pulled to opposite sides of the cell. Okay? It sounds an awful lot like anaphase. The chromosomes are going to start to decondense. The nuclear envelope is going to begin to reform. The chromatids are going to move to opposite sides of the cell. That sounds like anaphase. I'm hoping the next slide shows you anaphase 2. Check it out. It does. And by the time anaphase has been ticking away for a little while, the chromosomes have gotten more and more decondensed. The nuclear envelope is clearer and clearer and getting back into shape. The meiotic spindle is shorter and shorter. You softly, casually, gradually transition into telophase 2. And as part of telophase 2, you'll also have another cleavage furrow form for both of these daughter cells. They'll pinch across, separate the 2 chromosomes now into 2 separate compartments. And I worry, I'm not sure what I'm going to see next. An outline. You know what's going to happen next? They don't show you the big payoff here, but you started with a diploid cell. Because of a round of DNA replication, it became tetraploid. Then you went through 2 rounds of nuclear division of that tetraploid cell, and you end up with 4 haploid gametes. Finish off this cleavage furrow. This is a haploid gamete. 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 Look closely at what's going on, what's inside these haploid gametes. It's got a big blue one. It's got a medium-sized red one, and a small red one. Altogether, you'll find two big blues, 2 big reds, right? We'll sort it out in a minute. All right, but so you've made 4 haploid gametes. Meiosis I and meiosis II. There are just a couple of big differences between meiosis and mitosis. With mitosis, every round of nuclear division is followed by an interphase that involves DNA replication. With meiosis, you do 1 round of DNA replication at the start, and then you skip it before you enter into the 2nd round of nuclear division, meiosis II. Okay, so you reduce chromosome number that way. And the other difference is that these sister chromatids are staying stuck together, and there's this recombination stuff that's going on. And that's what crossing over is all about. We'll get to that in a minute. So what I just said is actually a pretty good overview of what this slide is showing you. And I can tell you what's going to happen next is let's take a closer look, a side-by-side comparison between mitosis and meiosis, and then talk about in words, I suppose, some of the differences between the two. So let's focus our attention on what's going on up here, and that's shown on this particular slide. So mitosis on the left, meiosis on the right. Let's talk about what's the same, and let's talk about what's different. With mitosis, you're starting off with a tetraploid cell, 4 copies of each of its 3 chromosomes. They line up on a metaphase plate, and then they get separated through mitosis to give rise to a pair of daughter cells that are each diploid. 2N means diploid, 4N is tetraploid, N is haploid. Look what you got in these daughter cells. You got a red chromosome 1, a blue chromosome 1, a blue chromosome 2, a red chromosome 2, a blue chromosome 3, and a red chromosome 3. 2 copies of each chromosome. It happens that half of these chromosomes came from this organism's father, and half of this organism's chromosomes came from its mother. But both of the daughter cells have exactly the same thing, right? There's no difference in terms of, it's not like you see 2 big blue ones over here and 2 big red ones over here. You get 1 big red, 1 big blue, 1 big red, 1 big blue, okay? So in mitosis, that's what's going on. You started out with a tetraploid cell here. You end up with 2 diploid cells. If you wanted to do this again, you'd have to have a round of DNA replication to make this daughter cell tetraploid, and then it would look like this, and you'd run it through this process. You can do this over and over and over again. Okay? That's what's up. That's what's going on in all of our somatic cells pretty much all the time. And it's also going on inside of our germline cells. However, also going on within our germline cells, and never within our somatic cells, is this other thing called meiosis. You start out the same. You start out with a tetraploid cell, right? No difference there. But at the start of meiosis, in prophase 1 of meiosis, you also have this thing called crossing over taking place at these chiasma. Let's make that a footnote for now. That's next on the agenda. We'll talk in more detail about that there. But this and this don't look all that different. In metaphase, for the first round of meiosis, all the chromosomes line up on a metaphase plate. Notice here that the non-sister chromatids are kind of clustered together. They're clustered together because of that crossing over that's taking place between them. Again, we'll talk more about that in just a few minutes. But they line up on a metaphase plate, and then there is a round of nuclear division that takes you from this tetraploid cell to two diploid cells. These diploid cells look different in an important way relative to these diploid cells. But you notice, aside from the little bits of blue and the little bits of red, you got a big red, a pair of big reds, a pair of medium reds, and a pair of blues, little blues over here. All right? And you have the reciprocal of that. You have a pair of big blues, a pair of medium blues, and a single pair of small reds. Here, you had an equal division of blues and reds, right? For every red chromosome, there was an equivalent blue chromosome. Equivalent here, fancy talk, homologous blue chromosome. Here, you got non-sister chromatids, so you still got two copies of the red chromosome, but they're sister chromatids. And the non-sister chromatid went to the other side. So there's an interesting, important difference that's happening because during metaphase 1 of meiosis, the sister chromatids remain attached. And then the other big difference between meiosis and mitosis is that you undergo one more round of division, but there is no DNA replication. And the end result is a set of 4 cells, 1, 2, 3, 4, each of which is haploid, and we call these things gametes. So these daughter cells of meiosis II could also be called egg or sperm cells. Here are words that draw your attention to some of these critical differences between mitosis and meiosis. I don't think these words on the screen right now say anything different than what it is I've just been saying to you. But if later on you want to take a look at the PowerPoint presentation here and you want to augment your notes with something that is doctrinally true because it comes from your textbook, this is the slide for you. Okay? So you can come back and take a look at this later if you think that would be of help for you. But it's really doing nothing more than trying to draw your attention to the similarities and the differences between mitosis and meiosis. This one shows it to you in pictures. This one shows it to you in words. All right. Meiosis 1 and meiosis 2. We're pretty much done with meiosis at this point. Mission's accomplished. The mission was I needed haploid gametes. I got them. Let's talk about this thing that's been sort of hanging in the air. I keep mentioning it and saying I'm going to tell you more about it later. Let's talk about crossing over. And I tell you guys, I'm working really hard to help you in this class, and I'm losing track of all the cool words that I've been given to you. Reticuloendothelial system, I think, is at the top of my list. I've been hearing from several of you that it's had the effect that I expected that it would. You know, feel free to share with me stories about your success with words like that. But I got to tell you, the single best word I think I'm going to give you all semester is up there on the screen right now, and I finally get to share it with you. I've been wanting to say this to you for a while because I know the power this word has. Synaptonemal complex. How about that one? You know what a synaptonemal complex is? It's what biologists use to describe what's going on, what's setting up to allow crossing over to occur. Crossing over only occurs in prophase 1 of meiosis, only at the beginning of meiosis. I've shown you some pictures that give you a sense of what's going on. I'm going to show you a picture now of an actual real-life synaptonemal complex in a little bit. Here, I'll skip to that. Where is it? Oh, man, there it is. All right. So there's an actual synaptonemal complex, a real-life captured on a picture, a crossing over event. Here it's called a chiasma. But that crossing over, the overlay of one chromosome on top of another chromosome during prophase 1 of meiosis, you know what's going on there is that the double-stranded DNA molecules of both of those chromosomes are getting broken, and there is an exchange of the DNA from one of those chromosomes with the DNA on the other chromosome, the equivalent, the homologous DNA on the other chromosome. So you're swapping, in this scenario, a little bit of blue chromosome for a little bit of red chromosome. And you're going to end up with a chromosome that's mostly red with a little bit of blue, and another chromosome that's mostly blue with a little bit of red. That's what's happening at a chiasma. What I've just described to you is called crossing over. It's also called recombination. That whole business gets accomplished. Here's that cliche again. How does that happen? What goes on? A protein does it. You know what that protein is called? It's not just one protein, it turns out. It's a complex of proteins. It's a synaptonemal complex. Synaptonemal complexes are not trivial. It's a very powerful word, has magical qualities when you're trying to impress people. But it's not a simple complex. There's a bunch of proteins that are involved. And funny thing, it takes a fair bit of time to set it up, to get all these proteins lined up in just the right way, to break the DNA. You know what? You don't break DNA molecules on a whim. Cells are really careful about protecting their DNA, so you got to get everything lined up just right. It takes a while to set that up. It takes a while to make a synaptonemal complex. What I'm building to is this punchline. It's not a joke, I'm afraid. But what I'm building to is the rate-limiting step for meiosis is prophase 1. It takes longer to do prophase 1 than all the rest of meiosis, just because you're waiting to have crossing over take place, to set up the synaptonemal complex, to resolve it, and to end up moving on to metaphase. It takes a while. It takes a long time. It can take a couple days for prophase 1 to take place because of what's going on at these synaptonemal complexes. Okay? It's not trivial. It's a big deal. It takes a lot of time. There's probably a good reason for it, or cells wouldn't be wasting that time and wouldn't have all this complicated machinery in place and maintained to be able to facilitate it. Crossing over, it's a big deal. Let's talk about it. All right? So here, I'm going to talk with you about crossing over. I've already introduced you to a synaptonemal complex. Before I even talk with you about synaptonemal complexes and their implications, let's talk about one of the neat things that happens just as a byproduct of meiosis. This should start to look familiar now. Here we've got a tetraploid cell. It's only got two different kinds of chromosomes, big ones and small ones. Okay, the medium ones are not part of the story anymore. You got a big one, you got a small one. Look what could happen. In meiosis, because these sister chromatids are stuck together with each other, you could end up with 4 different kinds of gametes. You could end up with gametes that have just blue chromosomes associated with them. You could end up with gametes that have just red chromosomes. And again, chromosomes that originally had come from this organism's mother, chromosomes that had originally come from this organism's father. That can happen, right? That's one kind of gamete you might make. But you might also make these kind of gametes, where you get a big one from dad and a small one from mom, or you get a big one from mom and a small one from dad. If you count up the number of chromosomes, the math works out all right. But check it out, this gamete is different than this gamete. They both have a big chromosome and a small chromosome, big, small, but this one has dad's big and mom's small, and this one has mom's big and dad's small. Remember, that can't happen. That doesn't happen in mitosis. In mitosis, you make a copy of the nucleus for goodness sakes. You made a copy, right? There's no difference between the two. Here in meiosis, we can end up with gametes that have kind of different mixes of chromosomes. They're not, all these gametes are not the same. In mitosis, all of the end products are the same. They have the same complementation of chromosomes. Here, you get the same mix of chromosomes, but you might have gotten mom's version and dad's versions mixed together in different combinations. You know why that's a big deal? I'm going to tell you why that's a big deal. I'm excited to be the guy that gets to tell you why this is a big deal. This is a big deal. Let me tell you why this is a big deal. I've been looking forward to this all morning. I've been wanting to tell you about this because I got the perfect analogy to share with you. All right? Let's say, I'll make this all about me. It'll be easier, right? Let's say that my mom has a gene that would give an individual a spectacular ability in terms of vertical leap, right? A 6-foot vertical leap. Pretty cool, huh? And let's say that my mom also, on the same chromosome that she has this gene for a, well, no, we'll put them on separate chromosomes. Let's say my mom, she has the 6-foot vertical leap gene on chromosome 1, and she's got the dribbling gene on chromosome 2. How about that? And let's just say that for whatever reason, while my mom has the amazing vertical leap gene on chromosome 1, she's got the really crappy dribbling gene on chromosome 2. My mom's not going to be much of a basketball player, right? Oh, man, if only she could dribble, right? Wouldn't that be swell? But then the good news is maybe somewhere out there, my mom's going to meet some guy like my dad who, while he's got the sort of run-of-the-mill vertical leap gene on his chromosome 1, he's got a really good dribbling gene on his chromosome 2. Wouldn't it be swell if in the lottery, the random assortment of these chromosomes, when they would come together, that my mom's good dribbling, or my dad's good dribbling gene might somehow get paired up with my mom's good vertical leap gene, and we've got now somebody who can both jump and dribble. NBA superstar, right? $1 million contracts. Life is going to be really good. If you could only ditch my mom's bad dribbling gene and my dad's mediocre jumping gene, if you could get rid of those somehow and get those two good ones, the really good ones together, that's going to work really well. That's going to be a happy organism. You know what? Meiosis, by having genes on different chromosomes and by allowing those associations between the chromosomes to get broken during meiosis, that's exactly what can happen. You see why this is a big deal? Now, for every good deal that's going to happen here, there's going to be a real loser in this process as well. Because check it out. Let's say this is the preferred combination. Great vertical, where is it here? Great vertical leap, good dribbling, right? Mom's got the big vertical leap gene on the big red chromosome. Dad had the good dribbling gene on the blue one. This gamete is a winner, right? Everybody wants this gamete. You're going to be a superstar in professional basketball. But for every one of these gametes you make, bear in mind, you're making one of these guys. This is the gamete that has bad vertical leap and an inability to dribble. See how that's going to work out? It sucks to be associated with this gamete. Life is pretty crappy for these kids in the schoolyard. They're the last to get picked for pickup basketball games. It's not going to be a happy existence. But boy, everybody wants to be this one, right? Natural selection and evolution isn't friendly, okay? Individuals who get this complement of genes, they're not going to be happy. There's a pretty good chance they're not going to be able to pass those genes on to another generation at least nowhere near with the same abundance as individuals with this combination of genes. That's how natural selection works. That's what's going on with evolution. It's given us an opportunity to break the association between bad genes and good genes. If those genes are on different chromosomes, because of meiosis, we can separate them and we can move on. That's what's happening just straight up with meiosis. Remember, meiosis is only happening for sexually reproducing organisms. And so I told you at the start of today's lecture, at some point I was going to try to impress upon you the fact that sexual reproduction is better than the alternative. You know what the alternative is? Cloning, right? We don't worry about cloning too much, at least right now, for humans. But there are plants that can grow that way. And there are other organisms that can grow that way. And there's certainly bacteria that don't ever undergo sexual reproduction. That's what happens with them. We have, because we can break those associations between advantageous genes and disadvantageous genes when they're on separate chromosomes, we have a much greater capacity, a much greater ability to shuffle our genes in a way that lets us get combinations that give us an advantage, that give some of us an advantage. And we pay the price of having to discard the combinations that are bad, but for the good of the species, for the good of the whole population, this is a great strategy. It's a winning strategy. The bottom line is that because of sexual reproduction, we can adapt to our environments much more quickly in a much greater variety of ways than if we were simply growing through clonal reproduction. So that's a big advantage. Let's talk again about what happens with crossing over, because with meiosis by itself, you separate the associations between chromosomes, right? There's no guarantee you're going to get just dad's copies of the genes, and no guarantee because they're on the same chromosome or different chromosomes. With synaptonemal complexes and with chiasma, what's happening in prophase 1 is the strategy that I just described to you is a great winning strategy. This takes it to the next level. Because with a synaptonemal complex, now you can actually break the association between genes on the same chromosome. Everything I've just been talking with you about, my mom's good dribbling, my mom's good jump gene was on one chromosome, and her bad dribbling gene was on another chromosome. Synaptonemal complexes crossing over allow you to break the association between good genes and bad genes on the same chromosome. So if breaking the association between good genes and bad genes on different chromosomes is a big deal, this has got to be at least as big a deal when you can break the associations between good genes and bad genes on the same chromosome. Because look what happens, just when you come right down to it, we're going to end up making gametes here. Sometimes the recombination won't take place, but when it does take place, you're going to make chromosomes that are sort of a hybrid. Big chunks of sequence that came from dad, little chunks here of sequence that came from mom, and we have what you can call a recombinant chromosome, a chromosome that doesn't look like either mom's or dad's chromosome. That's what dad's chromosome looked like. That's what mom's chromosome looked like. I've not seen anything like this before. I've not seen anything like this before. We can do this breaking of associations between genes on the same chromosome. It only happens in prophase 1, period. You only make that synaptonemal complex in prophase 1. That's it. Doesn't happen in prophase 2. It does not happen in prophase of mitosis. It only happens in prophase 1 of meiosis. And again, it takes a while to set up that complex, to get it done, to have it done right. It can take a day or two. It's the rate-limiting step for meiosis. For sexually reproducing organisms, it appears as if every chromosome must undergo at least one crossing over event during prophase 1. They must, before you can move on to metaphase. So it happens a lot. With human chromosomes, there are usually many crossing over events because we have some pretty big chromosomes. The further apart two genes are on a chromosome, should stand to reason, right? You would have figured this out without my telling you if you stopped to think about it. But the further apart two genes are on a chromosome, the better the chance there will be a recombination event between them. Genes that are right next to each other, what are the odds that a synaptonemal complex will form between them? Relatively small. Genes that are a long way apart on the chromosome, what's the chance a synaptonemal complex will take place or form between them? Relatively larger. In lecture a week from today, I'm going to talk with you about how you can use that insight to map genes along the length of a chromosome. It's a process, it's a trick that geneticists used called recombination mapping. You're looking at the frequency with which the association between genes gets broken. If it doesn't get broken very much, they must be close together. If they get broken a lot, they're probably far apart or they're on separate chromosomes. Okay? So, I think I've delivered, all right? I told you I was going to tell you why it was advantageous to have sex. I hope I've made the case for why it's evolutionarily a good thing to have. And I said a little while ago, at some point I'm going to show you a karyotype. I said it would be the last slide. Guess what? That's a karyotype. You know what you're looking at there? That's actually a picture of a real person's complete set of chromosomes. Humans have 23 different chromosomes. I haven't been lying to you when I told you that the biggest is called chromosome 1. The smallest is chromosome 21. They got them confused. And then look at that, those sex chromosomes. For the women, you can feel smug because look at that. You got 2 X chromosomes. X chromosomes are huge compared to the little tiny puny Y chromosome. If I were to ask you, what kind of person is this karyotype from? I would hope you would be able to recognize that that's got to be a male, right? There's an X and a Y. We'll talk more about that a week from today as well. Oh, actually, 3 lectures from now. A week from today, you're taking a test. All right? So have a great weekend. When we come back on Tuesday, I'm going to talk with you about Gregor Mendel, and we'll do a little bit of genetics and get you ready for that test next Thursday. See you guys next week.